and I lived by it and I did it or not. Because if you didn't and your life was just haywire all over, you will be the most dissatisfied person, right? So think about those things. And the value could be anything, right? It's about spreading love. It's fine, totally fine. That's what we need. We need, we need that. We need humanity, right? I keep on harping upon that. If you believe in these stories, if you believe in these movies, stick by them. Stick by the value that it's given you, right? And I'm sure there are more than one movies that you like and love and all. We watch movies, but we forget them, right? We all are like amazing, कितनी अच्छी मूवी है, right? At the end of the day, but वो जब वही चीज आपको अपनी लाइफ में implement करने को आता है, you actually don't do it. Okay? And that's unfortunate. So you guys have to be mentally strong enough to actually abide by that. Okay? Right? Don't we all love people who are honest? Right? Right? But tomorrow you get a government job where there is a bribing thing that happens all the time. You'll be like, what an amazing job, right? What an amazing job. I want that job. You want an IAS job, right? You want a job of a person who's powerful in the industry, powerful in the society. Why? A lot of times, and I'm not condemning anything. I know that this is true. I've lived here. I know every second of this, right? How this works. Please make sure that you live by those morals that you set out for yourself. Very important, very, very important. This is what makes you who you are. Again, remember, every animal gets their food, okay? You are a different kind of animal, right? So humans are different animals. That's what they've got their brains. Conscience is a big thing, right? Conscience. So remember, conscience is the thing that you should stick by. My mother said, I'm trying to something negative about my mother, but I'll tell you this because I think all of our parents do that. So one day, my cousin, at some point in life, I have younger cousins, they got a job of a labor inspector, and my mother was very happy. Everybody in the thing was very happy. Oh, she became a labor inspector. He also became a labor inspector. How cool. Now unki life set hai. You know this word, right? क्यों सेट है? How is a labor inspector different than a peon? How? क्यों सेट है लाइफ डिफरेंट? क्योंकि उसके पास 2 नंबर का पैसा आएगा, right? Right? Our society today is riddled with this, unfortunately. This will prevent you guys to meet your potential. This will prevent a lot of good people to meet their potential. We need to make a society that is not that way. Okay, that's unfortunate. That's wrong, and that the major point of that is everybody lives by money is the god, right? And you see so many movies where you will be like, oh, but you will condemn that thing, but we all live by that. Don't do that. Live by your morals. If you believe in something, live by your morals. There's morals that these movies have taught you, whatever else, that you set for yourself, right? It's most important. Do that. It's unfortunate, but I'm telling a very simple, very prevalent message, but this is so important, right? Honesty. Somebody said honesty, dishonesty. You do your experiments. You say, okay, this result was this. I set up a hypothesis. My hypothesis was that X does Y, and it turns out your experiment tells you X does not necessarily do Y, but does a little bit different than Y. But you still tell the world X does Y. Okay? You set up a wrong precedence. Be honest to yourself. Nobody else is watching except you yourself. Okay? Not a God, no nothing. You are watching yourself. Be true to yourself. Okay, that's the most important thing. I want you to know that is what will set you up. Okay, that is what will set you up for success. And success is not defined by $1 billion I talk about. Okay, success is defined by whether you are true to yourself. You are happy with yourself. Okay, and you'll only be happy if you meet what you set out for yourself, because in the process you'll lose yourself, right? And lots of movies show this. So do that. My favorite movie. What a gisapit answer it would be. My favorite movie is DDLJ. I'm a dyad romantic inside. No, it is because I like the guy. fighting for his love against the parents. I mean, he didn't fight. He could have run away with the girl, right? That's the only reason I like it, because he actually respected his parents. He tried till the last moment, but he didn't give up. He didn't give up, right? That's one of my favorite movies, because I was young. I was 10th grade. I was in, romance is everything, right? I'm not gonna lie to you. That's one of my, well, probably my first one, probably the movie I've watched most number of times. The movie that I really love is Swades, for obvious reasons, not because I want to come back or anything like that, because I think how small amount of innovation can actually change lives, and I am all about changing lives. I'm all about innovation. So Swades is my favorite movie. With my favorite actor, of course, I love Shah Rukh Khan for what he did, because of, not because of he does, he does because it's, the charm of the time I grew up in, and all of that. So that's that. But the movie has so much message. I mean, there's lots of great movies. You guys are talking about really, really good movies, but I liked it because I felt connected with it. Very simple innovation. He didn't realize. I mean, people like me. I mean, obviously he was in NASA. NASA is next to me in Houston, right? So that's a real story, by the way. That's a real life type of story. I think it, modified, but it is for real life. People, if we can bring change in the lowest common denominators, that's a negative word, but the people who matter the least to most people, right, to most of the world, that's where, to me, is a great influence. Because the guy came to a village, taught the kids, taught the people that you have to fight for yourself, think about small changes you can do, and did it, right? There was lots of such moments in the thing. And so there are lots of good movies. I mean, lots of good movies, but that's a movie I just like because Shah Rukh Khan was a very simple man, not this, superhero or hero or whatever. He was a simple man doing simple things. And that's what you do on a daily basis, right? I would like to do that for my country. I mean, I'm now an American citizen, but my heart is here. This is what I think of every day. I open Times of India before I open CNN. So, that's that. So, I actually have a company here, GIAN Therapeutics. I opened this only because I wanted to contribute to the best technology to India. That's my first company. So I actually believe in it. I believe in doing things here for Indian setting. That's a nationalistic thing, but I would say don't think about nationalism. I know some of you said that, but it's more about working for the lowest common denominator, which is the person who's the bottom of the hierarchy. Okay, if you do that, there's a great satisfaction. I mean, I don't know if it could be personal. To me, that's a great satisfaction. That's what I would like to do. Okay, all right. That's all the movies stuff. Let's talk science for some time till we have to go. Okay. So we didn't cover the DNA methylation profiling, so I thought we'd do that. This can be done in multiple different ways. The fundamental to multiple of these methods is bisulfite sequencing. I'll talk about that in a second. That has been there for decades over decades over decades. DNA methylation was found many, like over 100 years ago. I mean, I have Santosh send you guys an article. That's all historic perspective on DNA methylation, because why I had him send you that, it's not so obvious when you read the article, but if you think about it, the important part of that is that it will give you a perspective on how discoveries from day one to 100 years down the line make a difference, how they occur over time. The discoveries occur over time. They take a long time to happen. As the innovation happens, as technological innovations happen, we make more and more and more new discoveries, right? So it gives you sort of a time frame window, if you will. I mean, I don't know if you get it from it, but try to see if you can. The historical perspective on how one thing led to another, right? And to another. And that's what science is. Science is about not one person's glory. I know I keep on glorifying Nobel Prize winners, but it's not about one person's glory. It's about it's contributed from multiple people over time. So the whole field of DNA methylation that has actually, made a difference now in people's lives has taken 100 years to build. And that small thing that you do contributes to that. So the message there is, yes, we might think that this guy did this, but actually that guy alone didn't do it, right? No one person does things. In science, it has to be an accumulated effort. So think about that. Look at that paper, and you'll get it. So bisulfite sequence was discovered many years ago. So there are other methods. MEDIP is essentially a method where you pull down methylated DNA. I'll talk about it in a slide going forward, and then you can just do the library amplification and do your sequencing. Methyl cap is not used, so don't worry about these, but they use methyl binding proteins. So I told you about methyl binding proteins. You can actually be innovative and use the MBD domain and then essentially fuse it with something where you'll be able to pull it down, like, right? And they pull down all DNA that is bound by a methyl binding domain. So you're just using an endogenous system in a cell to pull down what you want to pull down. There are systems like that. So that's a good system. It has its own set of drawbacks. RRBS is reduced representation bisulfite sequencing. I'll talk about that. It is a pretty good method, actually. So a lot of people use that. It saves money, but also gives you a very good picture across the genome about what methylation does. Infinium is an array. This was used for the last 20 years or so. Lots of DNA methylation profiling in the field is because of this particular array made by Illumina. Again, depends on bisulfite conversion, but it's on an array. Earlier, the company is called Illumina. Before Illumina came up with sequencers, people used to have these arrays. Arrays were very like tiny ports where you would have essentially a complementary sequence to that DNA. So you could probe, like they had like 480,000 probes or something. So you could probe essentially lots of portions of the genome that had methylation on it. So you would know whether that binds there or not, or enriches there or not. And then validation is simply one. If you're focused on one gene, one locus, and you want to see whether this region is methylated or this gene is methylated, then you just look at that. And we'll come to that. These are all sequencing-based methods, the first three, as it says, right? So you can essentially pull down your methylated DNA, go to sequencing, make library, do the same thing of exercising of ChIP-seq, ATAC-seq, similar. It's on a different level. Methyl cap, again, here you use an antibody to 5-methylcytosine. In fact, there is an antibody to 5-methylcytosine that works really well. So that's a very good method. MEDIP is a very easy, very good method. Just the same as ChIP, but much easier because now there's no more chromatin. You're just taking DNA, pulling it down, and sequencing. This requires MBD domain, as I said earlier, and that's a little tricky. So very few people use it now. So don't even worry about it. Bisulfite sequencing will come to that, and then this is array based, and this is simply qPCR based. Okay, and then there is a series of bioinformatic analysis that one does, similar to what you've learned, and we'll talk about that. So bisulfite conversion is the basis of most methylation based methods, and the fundamental principle on this is sodium bisulfite. If you treat a piece of DNA with sodium bisulfite, it will deaminate C to U. Okay, so cytosine to uracil right there, but it will only do to the unmethylated cytosine. So very smart, very simple chemical. You just convert all C's to U's, make the complementary DNA piece, and just sequence it with a PCR or whatever, protect it. So it will remain C. So once you convert U in the next round of amplification will become T, right? Because U is an RNA normally, it's not on the DNA. So it will next round of PCR become a T. So in the original sequence there was C, right? And you see corresponding sequence is a T or a C. Very simple. So your computational method or your PCRs, etc., are all defined by that. Cool? Very simple. But we served the community so well over so many years. Such a powerful, simple yet very powerful method, right? So bisulfite sequencing then uses double-stranded breaks, double-stranded DNA, denature it, bisulfite treat it. Again, go back to what the treatment was. If you look at the C, where is that? Methyl C is C, other C is U, right? And then you PCR it up, like what we discussed in the previous slide. You can now directly take it for PCR sequencing. This is not so you can just do that, or you can design a primer that is specific to the site, okay? And then you amplify it and you see whether this does it or not. So for example, this is a figure from my paper that I did, what, in 2008. Again, I realize that you guys don't look at the papers a lot. So let's just, very simple experiment again. So here we have a piece of DNA and there's a P1 and P2 primer. Forget about P2 for a second. This R1 region is where I'm trying to look for methylation versus not. Front of this gene called NeuroD2. NeuroD2 is a very important transcription factor required for neural development. So what we did was we took this piece of DNA and subjected it to the bisulfite sequencing, which I showed you earlier. We cloned them. Once we know which DNA we found, we essentially cloned them. Okay, so we did 10 different clones. Pick the 10 different clones from the bacteria, and this is what it's shown. When there is an open circle, meaning that is not black, that means unmethylated. When there is a black circle, that means that's methylated. Okay, so in this particular figure, as you can see, wild type have unmethylated CpG island. This is a CpG island right here. I told you CpG islands are in right upstream of TSS normally. They are normally unmethylated. Okay, and in the control experiment, remember the experiments are as good as your control. A control morpholino is like something that knocks down in zebrafish. I told you earlier. You don't see any methylation, so controls looks just like wild type. WT is wild type, right? When we knock out our gene of interest, we now see methylation, 1 or 2. So AID and MBD4 in this paper we showed where the demethylases in what I told you earlier. If you have time, I have some slides from the paper I thought might be useful, but we'll see if that we have time. This is a chip experiment, qPCR. So I know I'm chipping my AID and MBD4 at this place. Okay. So essentially, when I say that, so IgG is my control. Again, IgG does not enrich, so I'm looking for fold enrichment. So I'm looking for fold enrichment of this on R1 over R2. Okay, very simple. Did you get it? So R2 does not get affected by this perturbation, whereas R1 does. Okay, so in R1 I see like when I knocked out these two, I see methylation occurring, meaning this was keeping these unmethylated. All right, and I can see that when I have this region, I see higher fold enrichment compared to IgG. Okay, that means these two proteins were actually sitting there, were actually binding at R1. Okay, and then demethylating it, and therefore when I knock them down, I actually see methylation occur. Does that make sense? You guys get it? Cool? Okay. I'm going to ask somebody to come up and explain this. Can do that. But how many people can do that? I know you two can do it. Who else? You guys are smart and you're not afraid of coming up here. Anybody else? All of you guys are smart. I shouldn't say that, but you guys are not afraid of coming up here. How many of you are afraid of coming up here and explain to me? Now nobody will come. Now there'll no be yes sirs. Why? Come on. Anybody wants to come up and say exactly the same thing I said? That would just mean to me that you were listening to me. Right? Do you want to come up? No? Why? Why are you afraid?
I'm just camera shy.
You're what?
Camera shy.
You're camera shy. Oh, because there is camera there. Oh my goodness. Is that the reason?
Come up and get
क्या बेइज्जती है बेचारे कैमरे की? You don't come to the camera. Okay. How many of you can do this? Nobody wants to do. Everybody's camera shy. Come on, you guys all make your reels and put it on Instagram or whatever. The क्या बोलते हैं उसको? TikTok and da. No, none of you do. How many of you do that? You do it. Okay, good. How many of you don't do it? You guys don't do it? Why? What will happen? Okay. All right. I hope you guys got it. I'm going to keep moving. I need to catch my flight, so I want to make sure that I'm done. All right, me-deep sick. I told you guys this earlier, right? You guys, how many of you got it? Hopefully, methyl, methylation, no methylation, methylation, right? An antibody, oh, sonicate and denature. You can sonicate and denature. You can sonicate and denature. What does sonication mean? Somebody's taught you this. Good. So sonicate, that will break the DNA in places, right? Then you will have methylation fragments, unmethylated fragments, or non-CG containing fragments. You use a methylated antibody using a 5-methylcytosine. You pull down the DNA, very simple. Now you got fragments that bind only to methylated, and then your fragment sizes. You can sonicate up to your what you like to do, of course, based on how much settings of sonication, how much wavelength you're setting, and what are number of cycles you're doing. Okay, so you can do that. Once you pull it down, you sequence that, and we have all heard about next generation sequencing, right? I'm told yesterday that you guys have a sequencer in this university now of some sort. No? They did. Go on, use it. Okay. Good. At least learn about it. So same thing. You can do this on the reduced representation bisulfite sequencing or whole genome bisulfite sequencing. The same thing. First two steps on this are essentially just library generation. Okay. So in the library generation, you do your digest. In this case, they're doing MSP1 digestion rather than sonication. Make fragments. MSP1 works on CCGG. HEPA2 will cut it. If internal CPG is not methylated, MSP won't cut it. MSP will cut it no matter whether it's methylated or not. It's methylation insensitive, whereas HEPA2 is methylation sensitive. The two enzymes, just pairs, work on the same site. Okay. You cut it, so you essentially make fragments where methylation is, right, or CG, so it's just going to cut right where the CGs. You ligate your adapters. You can see the ligation of adapters, and then you do the bisulfite conversion. So you do a PCR amplification. So whatever is methylated will remain C. What's unmethylated will become T, right? We got this? Simple, very straightforward. And then you just sequence it, and then your computational analysis will come, and he will figure it out how to map it, and where T is a T, C is a C, right? Whereas C remains a C or C becomes a T. C becomes a T, it's unmethylated. C remains a C, it's methylated. Very simple, right? Computational analysis tools go like this: input preference, input reach, pre-processing, mapping, MSC authentication, methylation notation, I'm not going to go into this. You can read about it if you want. I mean, if I were to take that class, that will take 2 hours, okay? Because, well, I showed you guys in ChipSeq. I've even given you the command lines, etc., that we use. So if you are interested in bioinformatics, you can look at that. And I'm always happy to talk to you guys later on. You guys have my emails, or you can get it from Santosh. I'm happy to talk to you ever, call anytime, okay? Since you guys sat through my class for 6 days, I'll take some time out. I'm just letting you know, I appreciate you sitting up because I can't teach the benches, right? Okay, good. 5-methylcytosine, that was simple. That was my goal, was to give you guys what does it mean. How do you go on and identify the methylated regions in the genome? You could do simple qPCRs. There are HEPA2 MSP1 based methods where you essentially digest the same piece of DNA with HEPA2 MSP1, and then you use a PCR primer to the end of it. Remember one trick always: TAC. Well, you guys don't do experiments, so it doesn't matter. But TAC will not. TAC is a DNA polymerase. DNA polymerase. Okay. TAC does not proceed if the last end is mismatched. Okay, that means what? You made your 20 base pair primer, it's complementary, right? But if the last one, 5' to 3', starts processing from 3', right? If the 3' end is mismatched, meaning rather than having a CG, if it's a TG, it's not going to go forward. Get it? How cool is that? That's a beautiful method. And then you will know your MSP1, HEPA2, your digest, non-digest, you can use it to go forward. Okay, things like that. You can do a lot of stuff like that. Okay, all right. Beyond 5-methylcytosine, there are other DNA modifications. And I think somebody will teach you RNA modifications in a couple of days. And you will learn all about RNA modifications, another beautiful world that has come up in last 10 to 15 years. So hopefully learn that, but my goal is to simply tell you that there are more modifications than methylcytosine. In lower eukaryotes, there are a lot more modifications. I'm not even going to that. And I think you guys can read about it. My goal was to make sure that you learn how to read, which you do, which you know. My goal was to make sure that you guys learn how to read papers. So if you can go read papers about lower eukaryotes, you do please. I don't know enough about that, so I'm not going to pretend again. But in the higher eukaryotes, you do see 4-methylcytosine, 5-methylcytosine. In RNA world, you see more. Okay, you start seeing more things. They actually change again the stability of the DNA helices, right? So that's one of the things, and that's just mostly because of their structure and where they fit in, how is the interaction between that versus the next base, and does that change the charge interactions and all of that. There are various reported functions. You can read about it again in different that range all the way from basic transcription to stem cell function to whatnot. I mean, there's tons of literature in this space. These things, although, are not very prevalent. Okay, so they happen very, very rarely. They might happen in specific conditions, like I said earlier, right? Some specific histone modifications or DNA modifications might get increased in a specific environmental stress condition. I told you the story about fasting, right? Fasting improves the K-hydroxybutyrate for us, right? So these specific stress condition, specific metabolic exposures will change the levels of these things. And that all depends on what enzymes are catalyzing this, and maybe the cofactor is much higher now, enzymes become active, or maybe that particular condition has increased the level of that particular enzyme now. So the enzyme is at much more quantity, so it's able to do that. So these things change upon environmental exposure. Excuse me. I'll end in next 2 slides, okay? For this lecture. And the one important one is 6-methyladenine. 6-methyladenine is, we looked at that, right, in the previous slide? Yeah, the right one. Essentially position at a 6th position, where is the amino group is. You can add a methyl to the amino group because amino group has open valency. And this is done by, these enzymes have become very powerful now, and you will see again more and more literature. I think RNA guy will tell you more about these things too. Metal L4, that is a 6-MA methyltransferase, and people find these in specific stem cell differentiation conditions where they become high and all, and that can put a CH3 on adenine. Okay, and there are other demethylases like ALK B family, that's a big dioxygenase family members that will directly remove these marks, and that can also do that. It can happen also because of the Dimroth rearrangement. It's one specific kind of chemical reaction of 1-methyladenine to 6-methyladenine. So it essentially is a conversion, or it can happen due to incorporation of pre-methylated adenines during DNA synthesis. So that's the way it happens. So there's catalytic activity, there's a conversion way of getting them, or there is just a, you have in your RNA, there's a lot more 6-methyladenine. That's why I said the guy will tell you. So when RNA gets catalyzed, this DNTPs will become available, and they can probably get incorporated during the replication. Very rarely, but they get incorporated. So that's how they get in there. So it's essentially the writer's eraser, the reader module. I don't know if people know 6-methyladenine readers. They probably do actually. These are, probably do, I can't remember, but I think they probably have discovered the readers for these particular marks. So this is a very active area of research. There's a lot more that needs to be done, and because when things are not very abundant, it's difficult to do research on, right? You can understand. So unless somebody finds when they become very abundant, then only you can understand their functions. And to ensure that people know the function of something, you need in vivo systems, right? You can't just do in vitro something and in vitro and say, okay, this thing exists, unless you're shown that this happens in human cells or any kind of organism, not just human, right? In life anywhere that happens, then you actually have proven that whatever you saw in vitro in a petri dish or in a test tube actually exists in real world. I mean, everything else is possible because it, right? Otherwise, it's an artifact of an experiment. And that's another big thing you should remember as you become experimentalists. Lots of things we read and present or, or we believe on are actually artifacts. They're really not there. So therefore, all the big papers, the good science includes doing both in vitro experiments and in vivo experiments, right? Okay? And finding specific new biology of things. What do I have on this picture? See, okay, this slide talks about that there is CG methyl. I told you all normal cytosine methylation happens in CpG context, right? 5-methyl cytosine context. And there is has to be a G followed by that.